Wonderful. So I'll sort of officially welcome everybody back. And the next talk we have in this bone session is from Professor Sue Burchill. Thank you for joining us, Sue. And so Sue is the Professor of Cancer Research at the University of Leeds here in the UK. And she established her research group in Leeds in 1992. And she has a real passion for translating scientific knowledge in clinical practice. And also over here in the UK, we have the National Cancer Research Institute, and Sue sits on that bone sarcoma subgroup. So she's very, very well placed to give us a nice introduction today to the kind of oncology and biomarker landscape. And this is an hour session, so we will run straight through, and then we'll take another half-hour break from the screen. So I'll hand over to you, Sue, and you can take it away.
Okay, thanks very much, Sorrel, and thank you very much for the invitation to come and speak to you today. It's a pleasure. I'm going to talk about some research that we've been involved in for a number of years. Research that's actually been done largely by a very talented postdoc in my lab called Liz Roundhill. You can see her picture here. The work that I'm going to talk about has been funded by a number of different charities, including Sarcoma UK, BCRT, Little Princess Trust and Ewing Sarcoma Research Trust. And without them and their funding, it wouldn't have been possible to do this research. So we're really grateful to them for that. I sort of thought, what, you know, how great to be invited, but what real qualifications have I got to talk to you, really? So I thought I should probably show a disclosures side. Now, usually when people have disclosures, they're announcing, you know, financial interest in companies, that kind of thing. Well, I have none of that. So my disclosures are quite small. But I am by training a pharmacologist, and then I did my training and PhD in the Northeast in Newcastle. And as Sorrel said, I came down to Leeds in 1992 because I had this wonderful opportunity to set up a children's cancer research lab. So I'm based in Leeds, which is approximately 300 kilometres north of London. My other disclosure is that actually I don't work on all bone sarcomas. I work particularly on Ewing sarcoma, and I've been working on Ewing sarcoma since 1992. We do some work on osteosarcoma, and we've started to develop models for other bone cancer types in collaboration with colleagues who I guess have seen some of the work we've done in Ewing sarcoma, and actually this could well apply to a number of other sarcoma types. So we're expanding what we're doing by using knowledge with others. My other disclosure is that I'm going to talk very generally about a strategy for identifying new therapeutic targets and biomarkers. And I've tried to be not mention too much of our own work. I've tried to focus on the strategy because I think it's a strategy that could be applied to any cancer type actually, but certainly to sarcomas because of their common origins. But there are one or two slides where I have mentioned specific biomarkers that I'm interested in, but one of the challenges for us is to compare our work with that of others. So we find the best biomarkers that are going to be useful for the clinic. So other biomarkers and targeted therapeutics are available, is my disclosure there. So I'll talk a little bit about soft tissue and bone sarcomas and why I think, although we spend a lot of time looking at the differences in these different tumours, I think for improving treatments rapidly, we maybe should start and try and find some of the similarities. As I say, what I'm going to describe is work that we've done on Ewing sarcoma, but it could be used in any sarcoma type, the strategy we've taken. And I'll go on and talk about some of the potential new treatments that we've identified and the biomarkers, and how we can use these to improve outcomes for patients, really. So I'm sure you heard a lot this afternoon about the pathology of soft tissue and bone sarcomas, and one of the big challenges is the number of subtypes that there are. So I think the last time I read a paper on this, it was over 100 subtypes. So that's quite a challenge to be trying to think about all those different cancer types. So soft tissue sarcomas are more common, probably 6 times more common than primary bone sarcomas. Although these tumours are quite rare in adults, there is a peak incidence in the sort of young adult age group. So they can account for 15 to 20% of malignancies in the under 25-year-olds. So this is we often think of these cancers as being rare, but if we look at them as a whole group, they are actually quite common, certainly in young people. So just sarcomas are related developmentally. So they're all of mesenchymal stem cell origin. So rhabdomyosarcoma, for example, arises in smooth muscle cells, and muscle cells arise from mesenchymal stem cells. But for the primary bone cancers, these are arising in structural cells that are derived from mesenchymal stem cells. So chondrocytes, osteoblasts, the periosteum on the outside of the bone, give rise to or are cells in which primary bone tumours occur. So chondrosarcomas in chondrocytes, osteosarcomas in the osteoblasts, which are actually the cells of the bone, and Ewing sarcoma that originate in the sort of soft tissue on the outside of the bone, but often present in the bone because they invade into the bone very rapidly. So these tumours are coming originally from the same cell, so there is some developmental similarities. Of course, the things that we focus on most when we're thinking about patients and treating patients are the differences between patients and how best to manage patients that have different characteristics. And clinical features are really how we currently decide how best we might be able to help patients. So in a very simplified way, if a patient presents with localized disease, then they would tend to have a better outcome than patients that present with metastatic disease. And this is, the first thing that, one of the first things that clinicians would begin to think about when they're deciding or discussing with patients the optimal treatments for them. So the extent of disease is important, and how those patients respond to current treatments is also important. So patients with metastatic disease, so the majority of patients who have metastatic Ewing sarcoma, which accounts for about 25% of new cases, would receive surgery, radiotherapy, and systemic chemotherapy. Localized patients might have radiotherapy and surgery alone, but for those patients that have the worst outcomes, it's a combination of these treatments, and particularly combination of the multi-agent chemotherapies that actually were developed back in the 60s and 70s and have been hugely successful in improving outcomes for patients. They target rapidly dividing cells. They're very non-specific in the sense that's their mechanism of action, and with that benefit for patients, obviously there's sometimes some side effects. So I think now we've used chemotherapy for so many years and maximized what we can do with chemotherapy. We really need to think about trying to introduce more targeted therapies, and I'll explain over the next few slides what I really mean by that. So if we think about patients and how they respond to chemotherapy in a very simplified way, if we think about cells and these blue circles, are Ewing sarcoma cells. They don't look anything like this, of course, but if we have patients who are treated with chemotherapy and the chemotherapy kills all the tumour cells, then we see complete responses and cures, which is of course what we want for all patients. But unfortunately for many patients, we might see after chemotherapy some response. Shrinkage of tumour and reduction in tumour bulk generally. But we do see later on recurrence, and so these tumours often come back. So we might see a partial response, and then later on the disease reoccurs. And actually sometimes treating again with the same chemotherapies doesn't improve outcomes. So this schema shows what we think is happening at the cellular level that reflects what's happening to those patients that have these partial responses, but then are not actually cured by current treatments. So the chemotherapy kills the bulk tumour cells, so the green cells here, the green circles, leaves behind a population of cells which they're called many different things, but these cells fundamentally have escaped chemotherapy, so they're drug resistant, chemotherapy resistant, and then they're also capable of self-renewal, so dividing to produce daughter progeny, so repopulating the tumour. And during this process, in response to chemotherapy, these cells can also change, which means they develop drug resistance. So even if they might not have had, these cells might not have been drug resistant in the first place, the green cells, they may well have changed in response to chemotherapy and become drug resistant. And so then now, this patients who have relapse or progressive disease are often the most difficult patients to treat, because now these tumour cells won't respond to the chemotherapies that maybe they would do initially. And many of you, I'm sure, will know about the study RECO, where people like Martin McCabe is the CI for that study, and looking to see if you can find treatments that might be able to treat this progressive relapsing disease. So our scientific hypothesis is that if we can isolate these cells that I've coloured grey and find a way to kill those cells, and then combine the chemotherapy, the drugs which actually kill these cells, with standard chemotherapy, so using chemotherapy and a targeted chemotherapy, then we should, we hope, be able to cure patients, or certainly cure more patients. We might even combine multiple targeted therapies, but at the moment, actually getting a targeted therapeutic into a clinical trial is quite a challenge. So this is sort of the idea that we might combine targeted therapy with chemotherapy, which does have a role in tumour shrinkage. So this is the strategy that that we've been working on for the last few years, to try and find new ways to actually treat Ewing sarcoma. I've drawn it as a linear line just because it's easier to explain in that way. But of course, the targeted chemotherapy could be given at the beginning, or and then the chemotherapy, or how you use these agents in actual patients is quite complex to model in preclinical models. But there's lots of opportunities to combine agents, I think. So this is our strategy. This has been our strategy for the last few years. What we have done is worked specifically on patient-derived cells. So with collaborators in different surgical centres around the UK, we've been fortunate to get samples from patients who've given consent, of course. So tumour samples, these have been sent to us in Leeds, and from these, Liz, who's a postdoc who works on this project in the lab, has isolated patient-derived Ewing cells, and she's managed to propagate these in tissue culture flasks. So what this means is we put them in a plastic dish, and we put them in some media and put them in an incubator that's at 37 degrees C, so we keep them nice and warm, and we give them some oxygen and air. And Not all these cultures will grow, but some of them will grow, and we're able to divide, the cells will divide, and we're able to bulk them up to do research on. And from these populations of cells, Liz has been able to do high throughput assays where she's taken these initial cells and identified those grey cells that I was talking about. So the cells that are capable of self-renewing and are chemo-resistant. I just put this slide in just to demonstrate why I think working on patient-derived cells is so important. This is a dendrogram which summarizes RNA sequencing data from a number of different patient-derived cell cultures and established cell lines. So that this is the whole RNA transcriptome of the cells here that has been summarized. And what you can see very clearly is that these patient-derived cell cultures are all clustering together. There's differences, of course, that come from different patients, but they're clustering together, and they're clustering independent of the established cell lines, the cell lines that have been in culture for many years. And it's because of these differences that I'm convinced we should increasingly be using patient-derived cells in our preclinical pathway to try and identify more effective therapeutics. So with Liz, what we've done in the lab is use these patient-derived cells in two different ways. One is to use them in a drug repurposing screen. So what I mean here is taking the cells into an assay and then putting on lots of different drugs that have already been designed and have been in patients. So we know a lot about their safety, we know how to deliver them. So potentially, if we can identify drugs that have already been produced and are already being used in the clinics to treat maybe other cancers or even sometimes other diseases, then we could fast track agents that are killing these Ewing sarcoma stem cells, stem-like cells, the grey cells that are responsible for the repopulation and the regrowth of tumours, so driving this process of progression and relapse. So that's one approach that we've taken. The other approach is to actually sequence them and look at the DNA and RNA transcriptome, and from this we of course have looked at potential candidates for drug discovery, but our primary aim here was to identify genes that are expressed very highly in the grey cells, so in the Ewing's cancer stem-like cells, that might be biomarkers, novel biomarkers, to identify those patients that have those Ewing's sarcoma stem cells early at diagnosis, and so that these patients might be offered treatments to target these specific cells in a sort of personalized way. This process of drug discovery is how probably as scientists we usually work to identify novel treatments. If you read the literature, you'll see lots of different treatments, candidate treatments have been designed and evaluated in the literature. And to actually identify a molecule, a drug target, and then develop a drug and take that into the clinic, into patients, is a very, very expensive and long process. I think, well, people sometimes say 10 years. I think that's a very optimistic approach. When I was reading to see what the sort of FDA would say, they reckon 36 years. So in anyone's lifetime, you maybe got a chance of doing one or maybe two. So it's hugely expensive. $2 billion, I think, was the figures I'd seen quoted, but it is also a very long process and not very efficient. So there's only about 6% of new drugs actually ever make it into patients. So that's quite a high attrition rate, I think. So these are the two approaches: drug repurposing to try and identify drugs that are already out there, we might be able to accelerate into the clinical trials to be evaluated in patients. and then the transcriptome analysis to identify novel biomarkers. So I thought I'd just show you the machine that we use for the drug repurposing screen. It's called an operator. And effectively what we do is we put the primary cells into plates. And here you can see some primary cells that have been stained with DAPI is the blue you can see, which is picking up the nucleus. That stains the DNA in the nucleus. And TOTO3 is the red you can see, and this is picking up or showing the cytoplasm. And this is just a typical image focused in on just 4 wells. In any one particular experiment, we run 960 wells. So it's quite a high throughput mechanism to look for reproducibility and to screen lots of compounds. And here you can see in this vehicle control, so this is being treated with a carrier that we use to deliver the drug. There's quite a lot of Ewing's sarcoma cells there. And then if we treat these cells with doxorubicin or vincristine or etoposide, at what are very high concentrations, I have to say, 10 micromoles, you can see there's some decrease in Ewing's cell number here in these grey cells that we believe are responsible for progression and relapse. So whilst doxorubicin might be killing some of the cells that are in the primary cultures, certainly not killing them all, and similarly for these other chemotherapeutics. So we've actually used this screening approach now to look at just short of 1,300 small molecules, and Liz has been doing an excellent job going through all that bioinformatic data that kind of screen generates. I wanted to spend a little bit of time on this slide. It's a bit busy, so apologies for that. But this just summarises some of the data on a few compounds from that 1,300 drug screen. This top table shows you down here, there's a number of different primary cell cultures here. And on the top here, we've got some different compounds. So here we've got some chemotherapies that I'm sure you'll all be familiar with these names: doxorubicin, vincristine, etoposide, actinomycin D. We've got YK4279, which is a very exciting drug which targets RNA helicase and prevents some of the activities of the EWS R1 fusions that are important in driving Ewing's. Here's some retinamides, so fenretinide and all-trans-retinoic acid, which are actually used in other cancer types. All-trans-retinoic acid, for example, is used in the treatment of children with neuroblastoma. And then here are a series of aurora kinase inhibitors. All these particular ones have been designed to aurora kinase A. There are 3 different kinds of aurora kinases, A, B, and C. And in Ewing's, the predominant types are A and B. So the way to read this table is where there's a white block with some stars in it, this means that there's some activity. So for doxorubicin here, you can see in 4 of the primary cultures, we're seeing some significant activity. And if there are more stars, it means there's more activity. So here, doxorubicin is really having some strong effects on these 3 primary cultures, but not very much on this, and nothing at all on these cultures here. So you can look at this as a distant overview. So where you see some white, it means there's some promising activity. If you look at the retinamides, they're having no effect at all, so that's really disappointing. But if we look here at these aurora kinase inhibitors, we're actually seeing some of these inhibitors are having activity in all the primary cells that we're looking at. I just sort of mentioned these concentrations of drugs used in this initial screen here in this table are quite high, so it's 5 micromolar. So this is the initial first step to see if there's anything, any activity at all that we might want to pursue. But really these particular compounds were are looking very exciting from our screen. So on the bottom of this slide, I just wanted to show you some of the more detailed information that we can get from this kind of screen. So on this, all the plots of the same kind of format. So on this axis, on the y-axis, XY, yeah, that's right. You can see increase in cell number. So up here, if there's a mark up here, it means there's a lot of cells. And here, if you look at this line, you can see with increasing concentration of, in this case, vincristine, you're seeing a decrease in viable Ewing sarcoma cell number. And this is the concentration of vincristine on the bottom here. So this is a low concentration of vincristine, and this is a high concentration of vincristine. So the plots are all the same kind of format. These blue lines are all are all cell lines, so established Ewing sarcoma cell lines. And you can see the Ewing sarcoma cell lines really respond nicely to vincristine. And these lines here that you can see, the grey lines and the black lines, they're showing, in the case of the black lines, the parental culture, so the primary culture that we established from the patient of Ewing cells. And then the grey lines are the grey cells, the self-renewing drug-resistant cells that don't respond to chemotherapy at all. And you can see vincristine, although there's some effects on the bulk parental cells, we're really only killing maybe 50–60% of the cells. If we look at some of these agents that I've identified here, so a couple of the aurora kinase inhibitors, here you can see we're seeing much greater kill. So this is increasing concentration. Look down here, we're talking about nanomolar amounts of drugs, so very sensitive or very low levels. And when we see sort of this dramatic decrease in viable cell number, really at concentrations that would be achievable in patients, we feel quite excited about the potential of this. But this is all just, this is all preclinical data. It's an example of what we can achieve with the patient-derived cells. I have to say we were very excited when we saw the data on aurora kinase inhibitors. And when we look back through the literature, in fact, there have been a number of different groups publishing on the role of aurora kinase A in Ewing sarcoma and how you might be able to use it as a therapy, but it's not being used in a clinical trial to be evaluated in Ewing sarcoma patients. There has been a study that was published, now let me think, it's about 2016, a phase 2 study looking at aurora kinase inhibitors in a group of different soft tissue, all soft tissue, it's certainly sarcomas. And unfortunately, the compound that was used and the design of the study, there wasn't any clinical benefit, or it didn't meet the criteria for taking the drug forward. But of course, that would be one of the drugs, and there are lots of different aurora kinase inhibitors out there, new aurora kinase inhibitors that are being developed with more specificity and more, less side effects. We're very excited about aurora kinase inhibitors at the moment in the lab. So just changing track a little bit. So I mentioned we'd use the primary cells to try and identify new biomarkers of risk. And so, of course, we use, as I said before, clinical features to categorize patients into low risk, if you're likely to do fairly well with minimal treatment and have localized disease, compared to patients that have got metastatic disease, so widespread disease, or maybe just metastatic disease from the tumor into one site. These patients tend to do much worse, because we do know that the characteristics that we use in the clinic at the moment, whilst they're obviously very important because these patients do worse, they're not telling us the full story, because about 30% of people diagnosed with Ewing sarcoma who are thought to have localized disease actually relapse. And so they experience the same poor outcome that's normally associated with patients with metastatic disease. And this is because we don't currently have the tools in the clinic to identify the 30% of patients with localized disease who are going to do badly. So actually, if we could find a biomarker that identifies at diagnosis those patients with localized disease who will do badly, who will experience the journey that a patient with metastatic disease has, then I think there's a good reason to potentially treat those patients in trials that are designed to treat high risk disease. Or you might look to identify different treatment combinations dependent on their specific biology. But thinking about, the outcomes for patients, there's obviously a need to improve the ability to identify the patients who are really going to have the worst outcomes as early as possible, so that they can begin to make some informed decisions about what treatments they want to have. So by using our primary patient-derived cells with the RNA-seq data, so this is where we've looked at all the RNA and the transcriptome, it's called, in each of the different patient-derived cells. We've compared the primary ES cultures, so this is the bulk cells, to the grey cells that I keep talking about, so the ESCSCs, the cells that we think are responsible for progression and relapse. And we've compared the transcriptomes of those two cell populations to identify any genes that are specifically upregulated in the ESCSCs. And then if we find, we were looking to find them and we found some, then having found those, see if they could be used to identify patients who are doing particularly badly or may do particularly badly with the current treatment that they're receiving. So this is a bit of a complicated plot again, so apologies for that. But what this is simply showing is on the bottom here, we've got the different cells. Each of these spots is a different RNA. And then we've got the actual decrease here, so fold change, decrease in level of expression compared to increase. And you can see in the middle, there's a lot of factors aren't changing. We actually found a lot of different RNAs between the primary cells and these cells that we believe are responsible for progression and relapse. So 561 genes. So that's a lot of genes to think about. But we are specifically wanting to find in this particular strategy things that are expressed at high levels in the ESCSCs. So I'm sort of on this side, things that have got large changes in expression and are also significantly different. And we've looked at all these that are circled and named here, validated the expression differences at the protein level, and looked at to see if these things are predicting outcome for patients at all. And this is an example of one of the markers that is predicting outcome. So this is Neuroxin 1. And I've actually shown you the data from the protein analysis, because of course it's the protein which at the end of the day has the functional significance. So just on the bottom of the slide here, there's a photograph of a Ewing sarcoma tumour that's got low expression of Neuroxin 1. So this is effectively a Ewing's tumour that has been cut. So we've got a slice of Ewing sarcoma here, and these cells that you can see where it's really largely you've got these big blue nuclei and not very much pink around them. So the pink is the cytoplasm, and the blue is the nuclei. This is typical Ewing sarcoma, small round cell histology. And what you can't see very much of on this slide here is brown staining. So if we look on this slide, I think you'll see really clearly there's lots of brown staining here. And this is another Ewing sarcoma, but what we're seeing here with the brown stain, which is actually something called DAB, which is picking up an antibody that is bound to this section and picking up expression of Neuroxin 1. So the antibody has been added to this slide, and we've put the DAB on, which picks up the antibody, but you don't see any staining because the antibody is not bound to the protein because the protein's not there. But here we've got lots of protein, and so we're seeing lots of brown signal. So in what is a relatively small group of patients who have localized Ewing sarcoma, we've been able to look at this, and I imagine you're all familiar with these sort of Kaplan-Meier plots that we use to look at survival. So this is a survival probability here, time to an event, so event-free survival, and along the bottom time in days. And what you can see is this line here, which is showing high expression of Neuroxin 1. This sadly represents patients who are not surviving, compared to this line here, where we've got low expression of Neuroxin 1, and these patients seem to be doing better. So this is quite, again, remarkable, I think. It is showing a wide separation of these patients who actually, with current tools in the clinic, are diagnosed as localized and would be managed as patients with localized disease. This is the similar kind of plot, but here we're looking at overall survival. So again, they look slightly different, of course, because this is about events and this is about overall survival. But you can see the expression of the Neuroxin 1 seems to be separating these patients that have got localized disease, but not really, who are doing badly, who could maybe benefit from more intensive therapy, or certainly benefit from more targeted therapy. So this is exciting, I think. We're currently doing a piece of work with some friends from the EEC, validating this in a larger cohort, and also comparing it to other biomarkers, other tumor biomarkers, to see if this is adding the information to other biomarkers, or if it's better than other biomarkers, if we need a combination of biomarkers to tell us as much as possible about the patients at diagnosis, so that we can help the clinicians and the patients make decisions about what might be the most appropriate treatment for them based on their biology and sort of what we might predict from the biology to be the outcome. So what I'm talking about here is something that I'm sure you've all talked about a lot in your meetings. We're talking about personalized medicine. Rather than giving patients the same treatments, we're talking about looking at the biology of individuals and classifying them on the basis of their tumor biology, in combination, of course, with clinical characteristics, because they're still important. This is to add to the clinical picture. It's not going to replace it. It's just to try and help the clinicians and patients. So rather than giving patients all the same treatment, how can we refine the stratification of patients? So maybe those 30% of patients who are doing badly, even though they're diagnosed with localized disease, should be regarded as medium risk. I don't know, maybe they should be high risk, because they seem to be experiencing the same poor outcomes as those patients that are diagnosed with high risk disease. So maybe it's false to put another category in. Maybe they should just be moved into this group. I don't know, but if we could if we have more information, and if these preliminary data, and I just must emphasize that these are preliminary data, research data, but if these preliminary data hold up in larger patient groups, then this will be a discussion that the clinicians will want to have, I'm sure, because this will change how we manage, or how they manage these patients with localized disease. And of course, the goal is then these patients with high-risk disease are given specific targeted therapy that's based on their biology. So we're looking to identify molecular profiles that would allow clinicians to better stratify patients for more effective treatments based on the needs of the patient and on the actual biology. So that's really where I was going to wrap up. I just put this slide in because I think we all need a bit of good news now and again. If we look back at survival for childhood cancer, now this is all childhood cancer, back in sort of 1975, about 10% of children survived cancer. But if we look now in modern day, over 80% of children survive cancer. So I think this is good news for children with cancer. It demonstrates that research works. The research that people have been doing, clinical research, laboratory research, has improved outcome for those children. We all know that outcomes for sarcoma patients haven't dramatically improved in the last decades. So, you know, there's a real urgency and need to move some of the science into the clinic. Of course, you have to be confident about the science and its clinical relevance before you can do that. But I just leave you with this thought, because, you know, the research can make a difference. It is worth investing in the research. That's the way I think we're going to be able to cure Ewing's, bone sarcomas, sarcoma patients. So just finish with a few thank yous, particularly to the patients who have allowed us to use their material, because actually without that material we wouldn't have been able to do these studies at all. And I firmly believe it's only by studying the patients and the patient's material that we'll really make progress. Of course, my collaborators in the surgical centres who've kindly sent material for us to work with, and Molly McNee, who's a PhD student funded by Sarcoma UK, and Liz Roundhill, a postdoc I've mentioned a few times, who's actually been working with me for many years in the lab, who's funded by Little Princess Trust at the moment. Everything that we do is a collaboration, really, where we are working with, you know, small teams. We're working with the EEC, which includes clinicians, scientists. We work across all the disciplines to try and design science that's going to be most benefit to patients, and tell the clinicians about our results, and they feed back to us, you know, what they find using our science. So it's a conversation that goes on, and I do think that it's important to have those conversations, because that's by using the science, understanding the challenges in the clinic, and particularly understanding the disease, and the people who understand that best, of course, are the patients, because they're the closest to it and the biggest affected by it. So it's a team thing. I'm just a little part in Leeds doing some research that we hope might be able to be a benefit to patients. Actually, I think the strategies that we've been using could be a benefit to many sarcoma types, not just Ewing's sarcoma. So I've wattled on long enough, I think, so I'll stop there. Sorrel, if that's okay.
Thank you, Sue, and thank you for a really excellent talk, a very clear